Hello all, welcome back to my channel, The Biolab Scholar. So in my previous video, I introduced you briefly to molecular biology and the tools of molecular biology. So today in this video, I will be starting with one such tool of my molecular biology, which is molecular cloning. So what is a clone? Clone means identical copy of something. Cloning means making identical copies of something. But in molecular biology, by cloning, we mean making identical copies of the gene of interest that we want to study. Because we cannot really read one single gene by one copy. We need thousands of copies of that gene because later we need all those copies to make our transformant organisms like maybe plants or any other model organism that we are working with. So only there are very low chances if we are working with very low copy number, there are low chances of getting the transformant. So if we have a higher number of DNA, there are more chances of getting the transformant. So for that reason, we want to make thousands and thousands copies of the gene of interest. So for making that, those copies of gene of interest, we first need to have the DNA sequence of our gene of interest. So for that, today I will be showing you not one, not two, three ways of retrieving your DNA sequence from the internet. So let's get started. So for this, I will be sharing my screen with you so you can see side by side how I am downloading the sequence. So I'll go in the the screen. Yeah. So for the first method, I'm using NCBI. How you can go to this is very simple. You go to Google and then you search NCBI, and the first link is your National Center of Biotechnology information. And when you open it, you can see all these options. So here you can submit your sequence, download your sequence, analyze your data and whatnot. So, but because we are interested in downloading our gene of interest, we'll go to this drop-down menu and we'll go to gene. So here we click the gene. And now if you know any gene name, like for example, I'm taking the actin gene. So I'll write down the name of the gene by and then enter search. So you'll get like lots of options. Here you get all the options. Homo sapiens, like mouse, what not, like whatever, or like whatever your model system is, you can choose your gene accordingly. So just for showing an example, I'm choosing the Homo sapiens. So let's click on it. So here you can see all the information about the ACT, this actin gene, that like what assembly they have used, what is the organisms, what is the reference sequence, is it reviewed or not reviewed, and what is the gene type. So this is a protein coding gene. And then if you want to download the sequence, the DNA sequence, you have to scroll down a bit and you will go in the genomic region, transcripts and products region. And here you can see just beneath the genomic sequence, this drop down menu shows you the information of the gene, that it's on the 14th chromosome, that it's on the chromosome 7, and what's the reference, assembly and everything. So because we need to download our gene of interest, we will go on the FASTA and we'll click on FASTA file, and then we just wait to open the FASTA file. And here is the gene of interest. So you can just copy and paste this sequence in your Word file, and you can work on it, like designing the primers and everything. So this is one way. NCBI is for all the organisms, like it is for the plants, it is for the animals, for humans, like everything. The next database that I'm going to show you is particularly for the Arabidopsis. So I'll go for TARE. So I'll search TARE on my tab, and then I reach here. The first option is the Arabidopsis information resource. I'll open it. So this is basically for all the biologists that are working particularly on Arabidopsis, because Arabidopsis is the most common model organism. So it's a great website for downloading all the Arabidopsis genes. So here I'll also again show you an example of actin gene. So not actin, I'll say maybe the CT1. You can download any gene, like you just have to type it here in the column, and then you'll get all the different options, and then you check the gene of interest. Like I searched for ACT1, so I'll go and click on this option. So here you can see all the information about the gene. This is the map view of to download the gene sequence, you have to scroll down a little bit and reach this sequence option here. So here you can see the full length CDS, full length genomic, full length cDNA. It all depends on different type of experiments, what kind of sequence you want to download. So if I say, for example, if I say I want the full length genomic, I'll click on the full length genomic. And here is your sequence. So you can see it's a pretty big sequence. It's almost 4 KB, but you cannot directly use the sequence from here, because you can see the numbering here. This will not work if you just like directly copy paste on your Word file. For a better format, you just have to send it to BLAST, and then you are going to get the sequence in this box without any numbers. So here you can directly copy, just copy paste in your Word file, and you are ready to go. The third option is my personal favorite, and why you are going to see in a while. So the third option is phytosome. I'll just simply search phytosome. Yeah, so we are in phytosome database. So I love this database personally because it includes all the plant kingdom organisms. Like here you can see Brachyplastid, Viridiplantae, there are embryophytes, angiosperms, eudicots, and later if we scroll down, you will also get monocots. And because I'm working on monocots, I find it like very convenient. So here all the monocots, like grass family, Brachypodium is the model plant for monocot, how we have like Arabidopsis for dicot. And then we also have maize and whatnot, like the whole plant kingdom is included in phytosome. You can also download the gene sequence of your interest by going specifically in the database of your organism, because all the organisms have their own databases for sequences, but I prefer to use phytosome because I find it like very convenient, and you will see why I'm saying that this is my personal favorite. So for example, because I just showed you the example of the ACT1 gene from TARE, I'm again going to repeat the same here. And now I'll show you what is the difference you get in retrieving the sequence. So here you can choose the genome by selecting a tree. You can either select from here. If I click this, if I click on this one, you are going to see that this genome is now selected. But I prefer to write it down to just like minimize scrolling down. So because I have to search for Arabidopsis, I'll write down Arabidopsis. And then you get all the databases from Arabidopsis. So I'm interested in Arabidopsis thaliana TARE 10. So I'll click on this. Your genome is now selected, and then you can go and find genes by keywords, or you can search by BLAST or the other options. So I'm interested in finding the genes by keyword. So I'll write down ACT1 and then press enter, and then just wait. Yeah, so here you can see all the options. So then you have to just check the information that what gene you want to download. And then because I'm interested in this acetyltransferase 1 gene, ACT1, so I go and click on the gene report. So here are two options. Either you can choose browse genome, which I don't want. I want the gene report because I want to download my sequence. So I'll go on G option and then I'll just click on it. And then here you get all the information about the gene. To download the sequence, you have to scroll down a little bit, and a little bit more. And then you reach on the sequences option. So here you can find the genomic sequence, transcript sequence, CDS sequence, and protein. And whatever you want to download, you can just click. So now I'll show you what is the difference between in the genomic sequence from phytosome and how we already see in TER. So if I click on this, you see that the gene is already characterized by color coding. So this green and light greenish color is the 5' UTR region. And then you have this bluish and light bluish color for all the exons. And this white portion are your introns. And then this dark purplish and light purplish color is your 3' UTR. So basically, this characterization helps you at the later stages of your cloning when you want to design your primers. It is like very easy for you to track where your exon 1 starts. So let's say this is the ATG site. So here your gene starts, like the coding region starts. So it shows like very precise color coding. So it becomes like very easy for you to that you don't have to look for where your gene starts. So I find it very convenient personally, and it has like helped me so far a lot. And it's the same for every other sequence. If you go to see cDNA sequence, this is your 5' UTR, this is all of your exon, and this is your 3' UTR. In the CDS sequence, you only get all the exons, and then you have the protein sequence. So it's like really fun. And then you can also mark your first exon proteins to your first exon. Then this is your second exon proteins. Then this is your second exon DNA sequence. So I find it very convenient, which has helped me so far. And I think it is like also very helpful to you guys. And even when you copy paste this whole thing in your Word file, you are going to get this color coding. So which makes it very, very convenient. Like I really, really personally recommend it. If you are a plant scientist, use Phytosome. So whatever plant kingdom organism you are studying, you are going to get the sequence from this database. So that's why this particular database is my personal favorite. So yes, that's all for today. And then I hope you find this video helpful. And in the upcoming videos, I'll be telling you how to design the primer and how to name the primer, because in molecular biology, we have a particular way of naming your primer, which are going to help you in organizing. So yeah, stay tuned. And most important, stay healthy, stay happy, and see you in the next video. Bye-bye.